Hi, I'm Professor Gwen Miller, and this is an online review for the Amylase Lab being held at the McKinney campus in fall 2019. If you hear anything during this review session or see something that you are unsure about or that might contradict what your lab instructor said, your lab instructor is always the primary person to contact. So anything that your lab instructor has told you, please take that over anything that you may hear here. If you have any questions, again, I encourage you to talk to your lab instructor. Before we get started, I just want to talk a little bit about what the lab practical will look like as you interact with the lab practical. The lab practical is 50 questions long. Every place in the lab, there's 24 places for students to sit, will be a station. When you come into the room, you will be assigned to start at a station, and at that station may be equipment, there may be pictures, there may be tubes of colors, there may be a microscope, and you will be asked one or two open-ended questions at that station. You'll be given about a minute and a half to 2 minutes at that station to answer those questions. After the end of your time, the entire class will then be asked to move to the next station. You'll have an opportunity to view each of the 50 questions once. Then at the end of the lab, your instructor will give you over a few minutes to go back through and see anything else that you want to review. We're going to go ahead and get started now. I'm going to begin by working off of a whiteboard screen to go through each of the units. If you have your lab manual, you may want to use this because that's the same thing that I'll be using as I go through the units. After completing the whiteboard section of this, we then will have time to look at a few images, and I will show you case a couple of sample questions. Thanks. Hi, let's get started with Unit 1. So on page 2 in your lab manual, you have a chart that looks somewhat like this. You should know everything about this chart and be able to convert within the metric system. For each of these, you should understand what the prefix stands for. So K stands for kilo, for instance, which is also the same as saying 10 to the third of the base unit. As we look here in the center, this is our base unit. We have 3 base units in the metric system. We have the meter for length, we have the gram for mass, and we have the liter for volume. With these base units, we can then add these prefixes to them and then convert between them. Be careful as you look at conversions out here on the smaller end, there are hidden units. So if you're converting from micrometers to nanometers, micrometers to nanometers, you're going to actually have to move it 3 decimal places. For example, excuse me there. If you're changing 53 micrometers to nanometers, you're moving that decimal place from micrometers 1, 2, 3 to the right. So you're going to do the same with your original answer. 1, 2, 3 to the right. 53 micrometers actually becomes 53,000 nanometers. So you should be really comfortable with this Unit 1 chart. Again, all of those basic metric system prefixes, they're what they stand for and how to convert between them. After that, on page 3, we converted from English to metric units. You should be able to use the equations as provided to convert from Fahrenheit to Celsius or from Celsius to Fahrenheit. On page 4, you then talked about converting English to metric with length, mass, and volume. As we provide different conversion factors, you should be able to convert between English and metric system. Exercise 1.3 on page 5 is the scientific notation. You should be able to take any number and put it into scientific notation and take any number that's in scientific notation and write in expanded form. As a reminder, numbers in scientific notation all fit the following form. They're going to have 1 digit in the ones column, decimal, everything else, times 10 to some power. So for instance, the number 23,032 becomes the following. If I'm taking this number, 23,032, and trying to turn it into scientific notation, I would rewrite it with the 2 in the ones column, decimal point, everything else, 3032, times 10 to some power. To figure out what power I'm going to use, we have to look at where the decimal point was originally and where it moved. So it moved from being right here, 1, 2, 3, 4 decimal points to the left this time. So our answer becomes 10 to the 4th, a positive 4, because this was a very big number. The next experiment that we did was exercise 1.4, where we became familiar with laboratory equipment. You should be able to identify all equipment used in this lab and in subsequent labs. We then talked about the difference between accuracy and precision. Let's imagine that a scientist is doing an experiment and the correct or the right answer is 100. So that's the answer that if they did the experiment right, they should get. When we talk about precision is not talking about accuracy. Precision is just talking about always getting the same value. So for instance, this individual got, when they ran the experiment, the answer is 52, 52, 53, 52. This individual right here that we're circling with the blue pen, they are very precise. They're getting the same value every time they do the experiment, but they're not very accurate. They're not getting the correct answer. On the other hand, we could have this individual that gets the answer of 50, 100, 150. You can see that their values are a little bit more spread about. If we average those, they're accurate, but they're not very precise because they're not very repeatable. So you should be able to look at a series of values and decide if they're accurate or precise. Again, precise means you get the same thing over and over again, and then accuracy means you're actually getting the correct answer. After that, we then measured items with a ruler. You should be able to accurately measure length with a ruler. You should be able to use a scale on page 8 as we measured mass. You should also be able to measure volume as we did on pages 8 and 9. In measuring volume, you should understand that the beaker and the Erlenmeyer flask are not very accurate. However, the graduated cylinder and serological pipette are more accurate because of their gradations, because they're able to be more precise. As we look at the graduated cylinder, make sure that you understand what the meniscus is. The meniscus is that little bit of a curve that we get in the liquid in the tube. With the meniscus, you should always try to measure from the bottom of the meniscus the volume. What this really means is that you need to be at eye level when reading the volumes. Exercise 1.5 is where we then use the micropipettes. You absolutely should be able to use a micropipette accurately. To use a micropipette accurately, first you set the volume, then you put the tip on, you press and hold the plunger to the first stop. At this point, you then put the micropipette tip into the liquid and you slowly release the plunger. This will slurp up the liquid for you. You pause a second, then remove the tip. You take this tip that's now full of liquid and put it into a delivery vessel. You press the plunger to the second stop, pause, and now the liquid has been delivered. At this point, you're going to release the plunger all the way, and you can eject the tip into a waste container. Some things to remember about micropipettes: you should never carry them upside down. They should also have no air bubbles. With the micropipettes, it's really important that you know how to select the correct micropipette for a volume and that you can also read the micropipette. So, and just to remind ourselves, the P1000 is named based on that largest volume. It's got the blue stopper up top. You're going to use the blue tips with the P1000. It runs with a red number followed by 2 black numbers. That first red value is actually the thousandths. And the middle number is the hundreds column, and then that final is the tens column. The P200, best at measuring volumes between 20 and 200 microliters, is going to have 3 black volumes. This is going to use the yellow micropipette tip. The first 0 reads the hundreds column, followed by the tens column, and then the ones column. The P20, also with a yellow plunger tip and also using the yellow tips, is going to have 2 black numbers followed by a red number. On the P20, you're going to first have your tens, and then your ones, and followed by the tenths. So for example, if we imagine the following volumes on a P1000, if it reads 0, 1, 2, that is the same as saying that we have 120 microliters. On the P100, if it reads 0, 1, 2, that's the same as saying 12 microliters. On the P100, if it reads 0, 1, 2, that is the same as saying 1.2 microliters. Again, you should be very comfortable and confident with your use of the micropipette. After completing the micropipette exercise, you should have figured out the following relationship. For water under standard condition, the following is true. 1 gram of water will always equal 1 milliliter of water. Or in other words, if you have 1,000 microliters, which is the same thing as 1 milliliter of water, that should weigh 1 gram. And you looked at this relationship as you explored that graph that you made. Statistical analysis, you should understand how to get the average value of any group of numbers. We did that in a variety of labs, but it was first introduced here. The average is calculated by adding all of your data points together and then dividing by the total number of data points. Unit 2 begins with some important terms that you need to be able to identify. You should be able to identify the independent variable and the dependent variable for any experiment. If graphed, the independent variable generally goes on the x-axis with the dependent variable going on the y-axis. You should also be able to identify a hypothesis and create hypotheses for experiments. In exercise 2.2, this is where you learned how to use the light microscope. You should be comfortable using a light microscope to find items on slides. As a reminder, the basic process of using the microscope is as follows. First, you place the slide on the stage with the stage at its lowest setting and the 4x objective in place. Then, while looking in the ocular lenses, you use the coarse adjustment knob to slowly bring the stage upwards. This is going to slowly bring the object into focus. Once you've found the object with the coarse adjustment knob, you will then use the fine focus to bring it into fine detail. At this point, you should not touch the coarse adjustment anymore. Once you've found the object using the 4x objective with the coarse adjustment knob and the fine adjustment knob, you do not use that coarse adjustment knob again. At this point, instead, what you can simply do is slide the 10x objective into place, use the fine focus to bring it back into sharp focus. Then again, just simply slide the 40x objective into place and use the fine focus knob again to bring that into fine focus. With parts of the compound light microscope, those were described for you in exercise 2.3. You should be able to identify all of the parts of the microscope identified on page 18 as well as their function. On page 2.4, we talked about microscopy concepts and principles. You should be familiar with all of those terms. Magnification is how much you enlarge the specimen. As we talk about magnification, it's important to remember what we mean when we talk about total magnification. Total magnification is equal to the ocular lens that you're using multiplied by the objective that you're using. So for instance, if I'm using the 4x objective because I'm just starting to look at a slide and I'm using the regular oculars, ours are at 10x, your total magnification is those two things multiplied together. So your total magnification is 10x times 4x or 40x total magnification. You should understand about resolving power, contrast, depth of field, and field of view. With resolving power, depth of field, and field of view, all of these change as magnification increases. So as magnification increases, your resolving power is also going to increase, meaning that as you zoom in on a sample, you're going to be able to see distinct entities as discrete units. However, as magnification increases, your depth of field is going to decrease. What this means is that what will appear in focus at a particular magnification decreases as you increase the magnification. This is also the same for field of view. Your field of view is going to decrease as your magnification is increasing. On exercise 1 on page 20, you looked at and explored this depth of field. You looked at the spirogyrus slide and saw how as we increased magnification, the depth of field decreased. With the spirogyra, make sure that you can identify the spiral chloroplast within that organism. Exercise 2.5 is where you practiced running through the steps of the scientific method. Make sure you understand the basic background informations, understand caffeine and ethanol, which one is a stimulant, and which one is a depressant. Exercise 2, we practiced making solutions. You should be familiar with the following equation: C1V1 equals C2V2. We can use this equation to calculate how to make other solutions. So for instance, let's imagine that I need to make 5 milliliters of a 3% glucose solution, and my stock is, however, at a 20% glucose solution. We can use this equation to figure out how to answer or make this new solution. So we plug in the values. My initial concentration that I'm starting off with is the 20% solution. I'm not sure how much 20% solution I'm going to need to use. My new concentration that I want is a 3% concentration, and my new volume that I want is a 5 milliliter volume. We then set this up and solve for x, and x turns out being 0.75 milliliters. So in order to make 5 milliliters of a 3% glucose solution, if my stock is from a 20% glucose solution, I would need 0.75 milliliters of stock. Notice, though, that isn't the only part of the problem. I am trying to make a 5 milliliter total volume. I'm going to need to use 0.75 mils of stock, but I'm also going to need 4.25 milliliters of water to dilute that stock solution down to the new concentration. So you should be comfortable using that equation and creating dilutions. In exercise 4, this is when we started looking at the heart rate of Daphnia. You should be able to identify the eye, the antenna, and the heart on a Daphnia. You should also be able to take the average value of a bunch of heartbeats to calculate the resting heart rate. Exercise 5, you then explored the effect of caffeine or ethanol on the Daphnia. With this particular experiment, if we give you a base heart rate, so let's imagine that the initial heart rate was 200 beats per minute of a Daphnia, you should be able to identify what a plus 10% increase or a decrease in 10% resting heart rate would look like. Given sample data, you should also be able to determine which concentrations of ethanol or caffeine cause that 10% increase or reduction in heart rate. Well, let's do Unit 3 now. In Unit 3, this is when we talked about how scientists don't only run experiments, they also use observations of the living world. We began in Unit 3 talking about the characteristics of life. You should be familiar with these characteristics of life that were identified also for you in lecture. So all living things are made out of cells, they have evolutionary adaptations, etc. As we look at the cells that we view in Unit 3, that's one thing to recognize, is that they do carry all of these characteristics of life. All the cells that we look at all have 4 things in common. This includes having cytosol, DNA, ribosomes, and an outer cell membrane. Despite all cells having these things in common, we can take all cells and divide them into 2 basic categories. These categories include prokaryotic and eukaryotic cells. Prokaryotic cells lack internal membrane-bound organelles. They are not going to include a nucleus. Eukaryotic cells, however, on the other hand, will contain internal membranous organelles, usually the largest of which is the nucleus, which is storing the DNA. We began class by looking at a variety of different eukaryotic cells. So that's where we'll start here. The first one that you looked at in exercise 1 was a protist. Protist is a junk drawer category. The protist is not a kingdom. However, the specific protist that we looked at was Volvox. You should be able to identify Volvox as being a protist and being a eukaryote. The next one that we looked at was the fungus Rhizopus. Specifically, you should be able to recognize that Rhizopus belongs to the fungus kingdom and what it means, and that this is a eukaryote. After that, we then looked at the Elodea leaf. This is an example of a plant kingdom, which is also a eukaryote. In order to identify the Elodea leaf structures, we did add a drop of iodine to stain the leaf. With the Elodea leaf, you should be able to identify the cell wall and the chloroplasts. Lastly, we looked at the animal cheek cell. In order to view this slide, we had to add methylene blue. with the animal cheek slide, since we are animals, not plants, we did not see a cell wall, nor did we see any chloroplasts. In using the microscope, we were able to review some of the concepts covered in Chapter 2. Again, you should be comfortable with all the parts of the microscope. You should also be comfortable with using the microscope, understanding which focus knob you use with which objective lenses. After we looked at those eukaryotic cells, we then turned our attention to the prokaryotic cells. So shifting gears now, thinking about prokaryotic cells. If you look at any prokaryotic cell, you should be able to identify it based on morphology and staining. Morphology is a fancy word which just simply means shape. So bacteria can be round, in which case we're going to call them cocci. They can be linear, in which case we call them bacilli, or they can be spirals, in which case we call them spirilla. If the bacteria find themselves in different groups, we can add prefixes to these basic morphologies. The prefix staphylo means that they're clustered like grapes. The prefix strepto means a long chain-like structure. In addition to morphology, we should also be able to visually identify if a bacteria is gram-positive or gram-negative. Gram-positive bacteria stain a dark purple color when using the gram staining protocol. Think royal purple or a grape jelly purple. Gram-negative bacteria, on the other hand, stain pink. This is indicative of a gram-negative bacteria. After identifying prokaryotes, specifically bacteria based on morphology and stain, we then discussed bacterial genetics. The following flow of information is common to all living organisms. DNA is used to create RNA. RNA is then used to create protein. Because this is shared by many organisms, what this means is that you can take DNA from one organism, transplant it into another organism, and you can still get a functional protein product produced. One way of doing this is specific to prokaryotes. It's called transformation. Transformation is where you take new DNA and insert it into a bacterial cell. That new transformed bacteria will then be able to create a protein that it wasn't able to previously. The specific transformation that we talked about was E. coli that had been transformed with a plasmid called P-AMI 2014. Some important things to remember about this. P-AMI 2014 is the name of the plasmid. It's the name of the new DNA that we are adding to the prokaryotic cell. If the transformation is successful and the P-AMI 2014, that DNA is able to insert itself into the E. coli, then that transformed bacteria is able to make the amylase protein. So amylase is a protein, P-AMI 2014 is the plasmid or the DNA that we transform the bacteria with. P-AMI 2014 does not only encode information for making the amylase protein, it also has ampicillin resistance. This allows us to select for positive transformants. With the transformation, you should be able to recognize what LB broth looks like when it has bacterial growth and when it does not have bacterial growth. When there was no bacterial growth, the LB broth remains sort of clear. When you get bacteria growing, it becomes cloudy. At this point in the lab, we then started talking about viruses. We really need to make a big break in our mind before we start talking about the viruses. Viruses are not cells. They are not prokaryotic or eukaryotic. If we think back to the very beginning, viruses are not a cell, or in other words, they are missing that first characteristic of life. Viruses do have evolutionary adaptations. They're able to respond to their environment. However, they are unable to reproduce on their own. They require a host. They're unable to regulate internal processes. They don't often contain enzymes. They aren't able to make their own ATP or process energy. They do not grow and develop. So viruses are these very small infectious particles. They're more than just a macromolecule, but they're not quite yet a cell. Understand what type of microscopes we use to view viruses, know their relative size range, and understand which characteristics of life they do not contain. In Unit 4, we begin by reminding you that proteins are made up of monomers called amino acids. Amino acids, when they link together, create a structure called a polypeptide, which is non-functional. That polypeptide has to be then twisted and folded to become the functional protein. The protein that we're especially interested in looking at for us is the amylase protein. In Unit 4, we are trying to figure out whether or not our transformed or non-transformed cells created any protein. In order to do this, we used a protein detection reagent called the Bradford reagent. This is a colorimetric test that reacts with some of the amino acids found in protein. With the Bradford reagent, if it's positive for proteins, it turns a bright blue color. If it has less proteins, or if it's negative for proteins, it contains a brown color. In order to analyze that blue and brown coloration, what we did was we were going to use a spectrophotometer. The spectrophotometer allows us to shine light on different solutions and see its absorbance or transmittance. Make sure that you understand the definitions of solutions, solutes, and solvents. Make sure you also understand how a spectrophotometer works. With a spectrophotometer, it can look at either the absorbance or transmittance. These values are inversely related. So if a solution has a high absorbance, that means that it's going to have a very low transmittance. Likewise, if a particular solution has a low absorbance, that means that it will have a very high transmittance. We can use the absorbance and transmission of light by the Bradford reagents to figure out how much protein is in a solution. If a Bradford containing solution has a high absorbance, this means that it contains a high amount of protein. If it has a low absorbance, this means it has a low amount of protein. In order to make the Bradford reagent work within the spectrophotometer, it's really important that we include a blank. One of the blanks that you were required to make in this lab included glass, water, and Bradford reagent. Each of these three elements does not contain any protein, but they will have an absorbance when you put them in a spectrophotometer. So what you did in the blank tube was you created this glass tube with some water and some Bradford. You put it in the spectrophotometer, and then you told the spectrophotometer, this is nothing. You asked the spectrophotometer to zero or to calibrate or to blank the machine. At this point, you could then look at your other tubes. Your other tubes contained glass and water and Bradford, but protein. In this case, when you put these other tubes into the solution, the glass and the water and the Bradford is going to be ignored, but you can focus on just the absorbance due to the protein. After setting up your blank tubes, you then had to set up a serial dilution. A serial dilution is where you systematically dilute a solution in a very systematic way. In this case, it was always diluting in half. We began by taking a substance that had a known concentration of 500 micrograms per milliliter. We then diluted it in half to get a 250 micrograms per ml solution. We then diluted that in half to get a 125 microgram mils solution. In each of these tubes, you knew exactly how much protein was present, and then you could put these in the spectrophotometer and figure out their various absorbances. Once you had your blank tubes and your serial dilution set up, you are now then ready to look at your experimental tubes. In your experimental tubes, you were trying to figure out how much protein was in them based on their absorbance. So for instance, you might have set up tubes A and N and got different absorbances, and then by using these absorbances values, you could then determine the amount of protein present. There are two ways to go about doing this. You can do a hand-drawn method or a Excel method. You should be comfortable with both. In the hand-drawn method, you make a graph where you graph protein concentration versus absorbance. As you plot your standard values, you might notice that some are linear and some are non-linear. In the Bradford reagent test, any absorbant values above 1 should be ignored. Also, any absorbance values out of the linear range should also be ignored. At that point, you then hand draw the green line, the serial dilution line, or your standard curve line. At this point, you can then take the unknown absorbance, in this case it was 0.492, find that value on the y-axis, draw a straight line over until you hit your standard curve line, then drop immediately down, As you drop immediately down, that point where it crosses the x-axis is the protein concentration. In addition to using the hand-drawn method, you can also use Excel. In Excel, you set up an XY scatter plot. It will again plot those same data points out for you. You should ignore any that fall out of the linear range or that are greater than 1. And you're graphing a protein concentration versus absorbance. The nice thing about using the Excel method is that after plotting this graph and having the computer calculate the standard curve line, that green line for you, you can then ask the computer to give you the equation of the line. Equations of a line always equal the form y equals mx plus b, where y is the absorbance and x is the protein concentration. After you are given the equation of a line, you can set the absorbance to whatever your unknown was. In this case, our unknown absorbance was 0.492. So you can set y to 0.492, and then you can solve for x to determine the protein concentration. In Unit 5, we wanted to see if our amylase protein was active as an enzyme. So some background about amylase is important to understand. Amylase is the protein that we've been talking about. It's also an enzyme, meaning that it can catalyze a reaction. Enzymes function by lowering the activation enzyme of a reaction. What enzymes do is they bind to a substrate and then break them into products. The substrate for amylase is a molecule called starch, which is a polysaccharide. So starch, in the presence of amylase, will break down into the product of maltose. In order to test to see if amylase activity is high, we can do this in 2 ways. One way to do this is by looking at our starch levels. The way to look for starch levels is by doing an iodine test. As starch levels decrease, or as the substrate level decreases, you will get a coloration change. When there's lots of starch, it will be black, which would mean that the enzyme was not working. When there's less starch, things will turn yellow, which means that the enzyme is working. You can also do a second test looking at the maltose, or the product. Maltose is a reducing sugar. One way to test for reducing sugars is to do a DNS test. In a DNS test, if maltose is present, it'll change brown. If it's not present, it will stay orange. So in this case, lots of maltose, lots of brown, means that your enzyme is active. The first experiment that you ran was a qualitative experiment looking for amylase activity in the bacterial extract. You set up a starch plate with the following 3 conditions. One was a negative control. This was just buffer. There was no enzyme. One was a positive control. This was your saliva. You were using your saliva because we know that human saliva contains amylase enzyme, which is very active. The third condition was the bacterial extract, which you were checking to see if it was active or not. When iodine is placed on starch, it will automatically turn black. Any areas on the plate that stayed black represented that there was no amylase activity. If, however, amylase was breaking down the starch, iodine would be breaking apart that starch. Amylase would be breaking apart that starch. So iodine would stay yellow. Areas where it stayed yellow on this particular plate represented areas where there was amylase activity. In exercise 2, you did a quantitative assay for amylase activity in the bacterial extract. You did this by setting up the four different conditions. Each of the conditions contained starch as the substrate and then different additives. In tubes A, you had starch plus the amylase buffer. There was no enzyme present, and so it was not expected that this would do much. In the second condition, it was starch plus saliva. Again, saliva was our control for known amylase activity, so we expected that that would probably do something. In tube C, this was starch plus maltose. Maltose is a reducing sugar, so this was a way to make sure that our DNS reagent was reacting to sugars. And then excitingly, we had tube D, which was starch plus our bacterial extract. In this case, we were not just looking at coloration of is it staying orange or are we getting a brown coloration, but we were taking these tubes and getting their absorbances. For most of you, the starch plus the buffer didn't do much of anything. This was pretty low absorbance. The middle 2 tubes, tubes B and C, gave you high absorbances, and then depending on your bacterial extract, you may have gotten some activity or no activity with the bacterial extract. Using these absorbances, you were then able to calculate exactly how much sugar was being produced in each condition. You did this by following the same methodology that we used in Unit 4. You set up a graph of sugar concentration versus absorbance of your known tubes. You then took the absorbances of tubes A, B, C, and D, and by using your standard curve, you figured out their concentrations. You may have also used the Excel method by plugging in your absorbance to figure out the exact sugar being produced. In this experiment, make sure that you can analyze what is positive for sugar production, what is negative for sugar production, and what is positive for the DNS reagent. At that point, you went to exercise 5.3. If you did not cover this in your lab, this was an optional activity. You should only worry about it if you covered it with your particular instructor. In this experiment, we were looking at the reaction that we know works. So we know that our enzyme amylase likes to interact with the substrate starch. We've seen that in exercise 5.1 and 5.2. What we were doing in this experiment was to see if perhaps we could get amylase to also react with a related substrate called cellulose. Cellulose and starch look very similar. Cellulose is linked together with beta glucose linkages instead of starch, which is linked together with alpha glucose linkages. So in this scenario, you set up the 4 following conditions. You had cellulose plus saliva, again, that's your enzyme, cellulose plus bacterial extract, and then you took starch plus saliva and starch plus the bacterial extract. In both of the solutions containing starch, so starch plus amylase or starch plus bacterial amylase, you got a pretty positive reaction. In this case, these tubes turned quite brown with the DNS reagent, showing that lots of maltose was being produced. However, in the tubes that contained cellulose, the enzyme was not very functional. These tubes stayed orange, showing that the enzyme was not able to break apart the cellulose as a substrate. This shows us that amylase is specific for starch, and it does not work on cellulose. In Unit 6, we wanted to look at factors that could affect amylase activity. As a reminder, amylase is the enzyme that we've been talking about in the past units. Amylase, as an enzyme, is a protein. This means that we have to remember about proteins. Proteins at the improper conditions, too high of heat, the wrong acid or base, the wrong chemicals present, can denature. This is when a protein loses its three-dimensional conformation or its native conformation and goes back to being just a polypeptide. So what we were looking at in Unit 6 was conditions that may affect that enzyme and make it less than optimal. In order to do this, we decided to focus on starch as the substrate. In Unit 5, we looked at 2 different ways that we could look at enzyme activity. In Unit 6, we predominantly focused on starch as a substrate. As a reminder, to look at starch as the substrate, the way we did this was with an iodine test. Iodine in the presence of starch will turn black. Iodine in the presence of starch being black is actually a negative result for us for enzyme activity. It means the enzyme is not working if everything stays black. If, however, there's starch plus amylase, the starch will begin to break down, and this will no longer stay black. This will turn yellow. The yellow or the clear colorations are positive. It's a good result. It means that our amylase activity is functional. The first experiment that we ran was to look at the effect of temperature on amylase activity. This is where we had the gigantic plate with all those little wells that you had to go back every 30 seconds and check to see what colors turned. Based on those visual pictures, if you are given a picture of results, you should be able to determine which temperature was the optimal for enzyme activity. Again, it's really important to remember that yellow is good. The quicker that a particular row goes yellow, the faster that enzyme is working. In exercise 6.3, you then looked at pH. Make sure that you understand some of the basic features of pH. pH is a measurement of the hydrogen ion concentration. As hydrogen ion concentration increases, that means that the pH levels are actually decreasing. So pHs that are less than 7 are considered acidic. They have high hydrogen ion concentrations. pH levels greater than 7 are considered basic. They have low hydrogen ion concentrations. Make sure that you know how to use pH paper to determine if something is pH. Make sure that you can also determine if something is an acid, base, or neutral. After doing this, you then set up another one of those series of reactions where you had all of the test tubes full of different pHs, and then you look to see what happened in your iodine test. Again, if you are visually given some of these results, you should be able to identify what the optimal pH is for the enzyme you're shown. In experiment 4, you looked at buffering capacity. Make sure you know that the buffer is something that maintains a set pH when acid or base is added to a solution. A good buffer is a weak acid-base pair. The specific buffer that we talked about was the phosphoric acid or the phosphate buffer system. In this experiment, we ran a variety of different settings. In one, we had starch, our substrate, plus water, and then we added either hydrochloric acid or sodium hydroxide, plus our enzyme amylase, and we saw what would happen. In the other condition, we ran starch, but in this, instead of having water, we added a buffer. We then took that starch and we exposed it to hydrochloric acid or sodium hydroxide. Then we added enzyme and we saw what happened. In general, the conditions where there was just water and there was no buffer, the high acid and the high base meant that the enzyme did not work very well. These conditions stayed black for longer. You should recognize that hydrochloric acid is the name of the acid used. You should also recognize that sodium hydroxide was the name of the base used. However, in conditions where we had the buffer added, everything was exactly the same. Starch was still in the presence of an acid. Starch was still in the presence of the base. But in these conditions, the amylase, because of the buffer being present, was protected. And so in these conditions where there was the buffer present, these ones started turning yellow or lightening faster, meaning that this enzyme was able to function better when the buffer was available. In exercise 5, we talked about cofactors. Please make sure that you understand the following. EDTA is not a cofactor. Cofactors are things like calcium or magnesium ions. We specifically used a molecule called EGTA. EGTA is also not a cofactor. EGTA is a substance that blocks calcium, preventing it from working with an enzyme. Make sure you understand figure 4 from your lab manual, what all those parts are. In this experiment, you looked to see if the addition of EGTA affected enzyme activity. Again, if we give you sample wells and coloration, you should be able to determine whether or not EGTA is blocking the cofactor, or in other words, you should be able to look at the results and see if whether or not a cofactor is required for the enzyme to function. With all of these units, make sure that you understand how to use the micropipette. We used it highly this day in lab. Also make sure that you understand the effect of pH, temperature, and cofactors on amylase. In Unit 7, we looked at diffusion and osmosis in a variety of scenarios. Make sure that you understand the definition of diffusion and you can predict movement of molecules. Make sure you also understand the definition of osmosis and you can predict movement of water across a semi-permeable membrane. The first experiment that we did was the diffusion experiment. In this experiment, we were trying to look at two different factors. We were looking at how both temperature and the size of the diffusing molecule might impact diffusion rates. In general, we saw the following relationship. As temperature increases, diffusion also increases. This makes sense because as molecules are moving faster, that's what temperature means. Those molecules are then able to move quickly through the substrate. With size, we saw the opposite. If something is smaller, it is better able to diffuse because it's able to fit through the substrate. The way that we looked at this was by looking at two different substances. If we give you the sizes of different solutions, you should be able to predict which one is going to move slower or faster based on size and temperature. In addition to looking at this, we talked about diffusion rates. As a reminder, rate is distance traveled over time. So you could calculate the diffusion rates of your various substance. If, for instance, at time 0, your initial size of your well had been 0.5 centimeters, so this is our 0-minute distance, and then at 20 minutes, it increases to 1.1 centimeters. The diffusion rate for that first 20 minutes is 0.6 centimeters divided by the 20 minutes. That would be our rate. If, however, in the next 20 minutes it moved from 1.1 centimeters to 1.8 centimeters, in the next 20 centimeters it's moved 0.7 centimeters in 20 minutes, and that is our new diffusion rate. So make sure that you can read data similar to the data that you can find in Table 1 in your lab notebook, and that you can understand the results. The next experiment, 7.2, was looking at the semi-permeability of membranes. You should understand that we use dialysis tubing. Dialysis tubing is plastic that has little teeny microscopic holes in it that allow water to pass but prevents larger molecules from crossing. You set up the following conditions, which I'm going to draw out for us right now. In the first condition, you had a bag full of just 1% starch and some water. Outside in the beaker, you had water with iodine. We know that water is going to easily move in and out of the bag, so really we're interested in finding out what happens with the starch and the iodine. In the second condition, you had your bag also with 1% starch, but in this case, you also included amylase enzyme. Then in the beaker, you also had iodine. As you first set up the experiment, you were just trying to see which molecules would move. In this particular experiment, what you saw is that iodine would quickly move into the bag, and so iodine stained the starch in that control group, and it became nicely black very quickly. However, in the second bag, we saw that although iodine was probably still going into the tube, this bag was not changing black. The reason for this is that the starch and the amylase were actually reacting, and so amylase was breaking apart the starch and creating maltose. You should understand what colors the bag went, what was moving, and then you should understand the experiment that you then had to come up with. We predicted that in the experimental group, maltose was being created, and that's the reason the bag didn't go black. But we wanted to confirm that this was the case. What we did was we then did a test to see if maltose was being produced in that experimental condition. The way to test for maltose was twofold. You had two options. Some of you chose to do a DNS test. You could test both the bag and the beaker to see if there was any reaction, or you could use a sugar test strip or a glucose test strip, which will also test for maltose. In this case, what we found when we looked for the sugar is that the sugar was not staying in the bag. That maltose was actually small enough of a molecule that the maltose was being found in the beaker solution, not staying in the bag. So we sort of had a couple of different things going on in this experiment that we wanted to note. First, iodine was moving into the bag. It was a small enough molecule that it could go from an area of high concentration to an area of low concentration. The second thing that we wanted to note was that in the experimental condition, the iodine was moving into the bag, but there was no black coloration happening. In the second, or the experimental condition, the starch had been broken down. The third thing that we really wanted to recognize is what was happening to the maltose. So the maltose, or the product of the experimental condition, what's going on over here with the green circle, is that the maltose was being produced, but then the maltose is moving out of the bag from an area of high concentration to an area of low concentration. In this experiment, all of this was diffusion. We're talking about iodine and maltose moving. We're just using the semi-permeable membrane to see how it's letting these smaller molecules, such as iodine and maltose, move, but starch, that large polysaccharide, is unable to move. In the next experiment, we looked at plant and animal cells in various tonicities. You should be able to recognize animal cells in various tonicities. You should also be able to recognize plant cells in various tonicities. So, animal cells in an isotonic solution appear normal shaped. Animal cells in a hypertonic solution become shriveled, or we can say that they are crenate. Plant cells, when they are in their normal condition, they are called turgid. This actually happens when they are in a hypotonic solution. Their vacuole is nice and big, and it's pressing all of the chloroplasts out to the outer edges. If, however, you place a plant cell into a hypertonic solution, it becomes plasmolized. What happens is the vacuole is actively losing water. So my vacuole is this little teeny thing here. It's losing water. That causes the edges of the cell wall to collapse. That's called plasmolysis. And all of the chloroplasts become centered on the interior. Make sure that you can recognize each of these cells and what condition they're in and the word that we use to describe them. All right, here are a couple review images that we'll briefly go through. This is not entirely everything that we might see. It's just to give you an idea of how things might be set up on the practical. So make sure you understand how to do conversions within the metric system. That's from Unit 1. Make sure you know how to convert from Fahrenheit to Celsius and Celsius to Fahrenheit. Make sure you can identify all the different lab equipment that we've used. As we looked at the various lab equipments, make sure that you understand where to look at them to find any markings on them that might tell you about their accuracy. We talked about accuracy versus precision. Again, precision is always being really close. Accuracy, however, is being close to the right answer. This individual over here is both accurate and precise. Here is a meniscus. This thing is 35, maybe milliliters. This would be 36 milliliters. This would be 37. So the proper volume here would be 35, 36, 36 and a half milliliters of whatever substance we were looking at. When using the rulers, please be careful. The rulers in lab are sometimes funnily labeled. This is an inch down here at the bottom, 11 to 12. 1 inch equals about 2.5 centimeters. Each of these individual lines is actually a millimeter. That large number 1 is 1 centimeter. The large number 2 is actually 2 centimeters. So be careful, be comfortable with centimeters and millimeters and understanding which is which. With the micropipette, make sure that you understand how to read the volumes. This right here must be a P1000. How do I know that? The top value is a red value. So in our lab, again, the top value on the P1000s was red, on the P200 all are black, and then on the P20s only the bottom value is 1. And you should be able to recognize this. This is 210 microliters for the P1000. This is 21 microliters for the P200, and this is 2.1 microliters for the P20. Here's the microscope. Again, make sure that you know all these different parts, what their labels, what if we labeled them, and also their functions. When viewing a microscope image, we always begin with the 4x objective, and we use the coarse focus knob first, followed by the fine focus knob. Then when we go up to higher objectives, the 10x, the 40x, the 100x objective, we're just using the fine focus knobs. To figure out total magnification, it's the power of the oculars times the objective for our total magnification. The diaphragm is this lever down here that allows us to adjust for contrast, and it's going to allow us to regulate the amount of light that goes in or out of a substance. This was what the Spirogyrus should have looked at. This green helical structure that you see in here is a spiral chloroplast. What we're looking at right now is the effect of depth of field on a substance. So you can see that part of the substance is in focus, but part of it is not. And this is because as we increase in magnification, the depth of field decreases. Here's the water flea that we use, the Daphnia. You should be able to recognize various elements. So we've got our antenna, we've got our eye. The heart was this smaller structure back here. These were some maybe eggs. This will also move, but this is not the heart. Those are some of the respiratory structures. Here is an example of how you were seeing how various things may affect heart rate. So here we've got a set curve. This is how different concentrations of acetylcholine affected heart rate. If we gave you a standard heart rate, maybe we said resting heart rate was 100, and then we said what concentration would cause a 10% reduction. So if resting heart rate was 100, A 10% reduction in that would be 90. We could read over and we could see, oh, 90 heart rate came at about a 2.1% acetylcholine solution. So make sure you know how to read this type of information that you developed yourself in Unit 2. If you ever see substances that looks like this is not an actual organism. This is what a glass slide looks like. So please, please, please remember that you're close, but you're not there yet if you see this particular image. This is the Volvox. This is our protist. This is your fungus, Rhizopus. This is Elodia. These are the animal cells. All 4 of these are your eukaryotic cells that we observed. All of these contain internal membrane-bound organelles. This is an example of some of our different bacterial morphologies. So you can see here are the cocci. Because they're dark purple, we would see that these are gram-positive cocci. We've got some pink bacilli back here. These would be gram-negative bacilli. And again, if you see those spiral shapes, those would be spirilla. Staphylo and strepto are added to those terms based on whether or not things are arranged in clusters or chains. We looked at a transformation. Normally, E. coli has its own genetic information found in the nucleoid region, and this would grow up great in LB. LB is a growth medium. What we did was we took E. coli and we transformed it with a plasmid called P-AMI 2014. The plasmid P-AMI 2014 included a gene that allows it to produce the protein amylase. It also has a gene that encodes ampicillin resistance. So this particular transformed E. coli will be able to grow up in the presence of ampicillin. Normal E. coli would not be able to grow up in the presence of ampicillin because this is an antibiotic and it would normally kill the E. coli. Here's an example of our Bradford reagent. Again, you should remember what colors blue versus brown mean and which ones contain the protein versus don't contain the protein. If you're given a sample standard curve like we have here, you should be able to figure out the protein concentration of an unknown solution. So for instance, if you have an unknown with an absorbance of 0.5, you go up on the y-axis till you hit 0.5, you then come across till you hit the standard curve, then you drop down where it crosses the x-axis is how much protein you have, which in this case looks about like 13 mg per mil of protein. You should understand this reaction. Starch is the substrate. It's a polysaccharide. In the presence of amylase, it's able to hydrolyze the amylase into the products of maltose. This was an example of somebody's starch plate. You can see areas where the starch was stained black, areas where there was amylase activity went white. Here is the difference between a positive and a negative iodine test. So here we've got lots of starch present, meaning no amylase activity. Here we have less starch present. We're getting just the iodine color, so that means amylase activity is quite high. This is an example of a DNS reagent test. Here we can see something with high amount of reducing sugars going down to something with less reducing sugars. Again, in this test, we're looking for the increase in product. Enzyme specificity, this is starch versus cellulose. Both are made out of glucose monomers. One's made out of alpha linkages, the other one's made out of beta glucose linkages. What we saw is that amylase preferentially wants to hydrolyze starch. It does not like to hydrolyze cellulose despite the similarities in structure. Given a pH paper, you should be able to figure out what the pH value is. So in this case, oh, it's green. This is green. That's pH 8. Given the wells, you should understand what the light versus the dark coloration means in relation to enzyme activity. Again, light color means the enzyme is active. We're getting rid of the starch. Dark activity means that the enzyme is not active. We're not getting rid of the substrate. Here's a sample plate that I gave you here, looking at enzyme at various conditions, asking which is the optimal temperature. In this case, it would be the 35-degree situation, because here in this particular condition, we see that the coloration goes lightest the fastest, whereas in 25, 45, and 55, the enzyme is lagging behind. It doesn't go yellow as quickly. Here's some example EGTA values. You can see that when there was no EGTA, the enzyme worked very well. At low concentrations of EGTA, the enzyme was still working, but at high concentrations of EGTA, the enzyme stopped working. What does this mean? This means that EGTA is blocking the cofactor. When EGTA blocks the cofactor, it's not working anymore. Meaning this enzyme that we see on this plate does require a cofactor to function. Here's an example of the diffusion reaction. You should be able to measure the diffusion rates. Again, we were looking at the diameter of how far apart the dye was spreading over time. Remember that rate is calculated as distance over time. Here are some examples of our bag and beakers. In one case, the bag turned dark black, showing that the iodine was moving into the bag. In the other condition, the bag didn't go black because the enzyme had broken down the starch. And in this second condition, we wanted to see, is that broken down starch, is that maltose being stuck in the bag, or is the maltose getting back into the beaker? And in this case, what we found, same picture as before, is that when we had the starch with the enzyme, the maltose was able to get out of the bag into the beaker. Here's some images of animal blood cells. Here we have one that's nice and isotonic. This is our normal blood cells. And here we've got some blood cells that are in more of a hypertonic solution. You can see that the outer edges are no longer nice and circular. We're getting more of the squiggly, weird shapes. So this is a hypertonic solution with shriveled or crenate blood cells. Here are two pictures of plant cells. In our first Elodea cell, this is the normal condition. We say that these cells are turgid. They're in a hypotonic environment. That means that the vacuole is so big, it's pressing the chloroplast out towards the edges of the plant cell walls. On the right-hand side, we have plant cells that have been found in a hypertonic solution. This is the 20% salt. What's happened to them is that their vacuole has shrunk. As the vacuole has shrunk, the cell membrane has ripped off the cell walls, causing plasmolysis to occur. We call these cells plasmolyzed. This is what happens when you put plant cells in a hypertonic solution. All right, hopefully that gives you some ideas, and good luck studying. As before, if you have any questions about any of this material, please contact your lab instructor. These reviews went very quickly. If I accidentally missaid something, please double check before you study it the wrong way. Your lab manual, your lab instructor is always the best source of this information.